Right, so hello, Northwithwa, Acrosa, Vivoscal Keel. So good evening, everybody. Thank you very much. Good evening, everyone, and welcome to Kiel, and apologies to Heidi and her family for butchering the beautiful Welsh language and my pronunciation then. So my name's Christian Mallon, and I've got the privilege of being Executive Dean here in the Faculty of Medicine and Health Sciences at Kiel University. Tonight we welcome Professor Heidi Fuller to deliver her inaugural lecture. Inaugural lectures are a key part of our academic timetable, giving us the chance to celebrate the success of our team and to understand more about the work and the activities that they do. Originally from North East Wales, Heidi received her first degree in biomolecular science at Bangor University in Wales, before undertaking an industrial-funded PhD in molecular biology and biochemistry. Following a period as a postdoctoral researcher in the NHS, Heidi transitioned to academia here at Kiel in 2013 and was promoted to Professor in Medical Sciences in 2024. During her time here at Kiel, Heidi's held a variety of academic leadership positions, including being the Faculty Dean for Education. She did this during COVID, which is a really busy and difficult time, as you can imagine. Heidi's led work around our strategic priorities relating to the development of new educational programmes to really meet the growth that's needed in the NHS, and has also led on quality assurance, educational enhancement, and internationalisation of programmes in global and public health. As the head of the newly formed School for Allied Health Professions and Pharmacy, Heidi oversees education and research programmes spanning diverse disciplines, including pharmacy and pharmaceutical sciences, allied health therapies, medical technologies, and active living and sport. Alongside all of this, which is a big job in itself, Heidi is the Laboratory Research Director at the Wolfson Centre for Inherited Neuromuscular Disease, a Muscular Dystrophy UK Centre of Excellence based at the Robert Jones and Agnes Hunt Orthopaedic Hospital, one of our close partners here at Kiel. In collaboration with clinical and international researchers, her team are using cutting-edge molecular, genetic, proteomic, and bioinformatics tools to understand disease mechanisms and to identify novel proteins and pathways that can be modulated through drug therapy and genetic-based treatments. Heidi also holds several external roles, including being an Associate Non-Executive Director at the Shrewsbury and Telford NHS Trust. Alongside that, she's decided she needs to diversify what she does because she clearly doesn't do enough. And so Heidi's taking on a new role as an expert reviewer of healthcare systems and has so far worked with the Gibraltar Government, the Falklands Island Government, and the Ministry of Health, Tourism and Sport in Anguilla. On a personal note, Heidi's quite simply a wonderful colleague. She's intelligent, caring, and could be described as being bendigedig. She shares her life with 4 men who she adores. Her wonderful husband, Monty, along with their 3 rescue whippets, Maurice, Vincent van Rasterbaan, and Professor Huxley. I think if there was ever a question that was waiting to be asked at an inaugural lecture, it's how Heidi decides to name her pets. So please join me in welcoming Professor Heidi Fuller to deliver her inaugural lecture.
Diolch yn fawr, Christian. Thank you very much. And thank you, everyone, for coming along this evening. So the title of my lecture today is Genomes Without Borders, Personalised Medicine Across Global Health Systems. In choosing this topic, it was a little bit difficult for me because my research pathway and my education interests have been fairly separate, I think, until recently. And so I've tried to find a theme that I think hopefully brings everything together. I won't spend long describing my academic journey. I think Christian's done that justice just now. But just to point out that I originally joined Kiel in 2013 as a research fellow, but I had quite I suppose, a slightly unusual pathway into academia in that I held that role alongside of a teaching fellow role that I was given in 2014. And I still to this day remember Alwyn interviewing me up at the CEC when I joined postgraduate medicine. And then I finally managed to convince Kiel to give me a proper job, a full-time permanent job as a lecturer in 2016. So this is me in GCSE Science in Llangollen in North Wales, obviously not yet trusted to hold a pipette. I asked Copilot, an AI piece of software, for those of you that don't know, for a funny Welsh word related to science. And instead it decided to make up a word. I said for Genesis, which I thought Christian might appreciate. And the definition apparently is the biological or cultural process by which a person becomes so Welsh that it spontaneously starts composing poetry and singing in a bardic competition. So yeah, I think that one's a keeper, and you heard it here first. So I then went on to do my undergraduate degree in Bangor, and in my second year I had the great privilege of being awarded an O.V. Jones scholarship. O.V. Jones was a local obstetrician and gynaecologist, died long before I started my degree. And little did he know that he would have changed probably my path, because I sincerely doubt I would have gone into research and academia had I not have had that opportunity in my second year. Here's us in the lab. Next to me, sadly no longer with us, Professor Mike Dernhoff. He was my supervisor in the second year and for my third year project. So we researched parasite infections and the link between specifically schistosomiasis and allergies. Mike was quite a character. I remember not long after I first met him, he came to pick me up in his Fiat Panda with several bags of manure on the back seat. Very down-to-earth character, but had a great role in shaping my journey, as did Stan next to him, who taught me much of the fundamental science skills that I have now. So moving my way up the food chain, I then went to do a PhD in allergies, specifically shellfish allergens, with Professor Glenn Morris. We were part of the University of Wales, based in Wrexham at the time, and then transitioned over to the Orthopaedic Hospital in Oswestry. So here I developed an immunoassays, an ELISA and a lateral flow-based device in collaboration with a company to detect hidden shellfish allergens in food. And these were sold commercially for a number of years. It was here that I developed an interest in proteomics technology. I actually remember when I first started with Glenn, and he said, "Do you like fixing things?" So I said, "Yes, I'll give it a go." And he said, "Good, because we've got a few machines that could do with some work." And there was one particular example. Actually, most people would have just gone out and bought a new piece of equipment, but there was a particular thing that had got blocked up. So I had a look online about how to fix it yourself, and went along and said it does carry a bit of a high explosive risk, the process involved. And I was told, just open the door and stand back a bit. So I did, of course, do a full risk assessment, and we all lived to see another day. I then moved to, I stayed on, but stayed in the NHS then to do postdoctoral research. And here I moved on to working on inherited neuromuscular disease with Glenn Morris and the team. It's lovingly called the Hospital in a Field. As you can see from the picture on the right-hand side, if you look on Google Earth, it is literally a hospital in a field. And here's some of our team on the right throwing axes, as you do in your spare time. So before I talk about my research and education endeavours, I just wanted to share a little bit about my general philosophy. I think this applies very well to laboratory research, but it's also something I try to encourage students with as well. And it's about the role of failure in discovery, or failing well. And any excuse to use any of my hobbies as a way of making an analogy. So those of you that know me quite well will know that one of the things I like to do is metal detecting. You typically only ever see pictures of the nice things I find. So a nice medieval silver hammered coin in a Roman fibula. The reality is this probably only crops up once every year or so, and most of the time I get up at 6 o'clock on a rainy Sunday morning and I dig up antibiotic tubes, mobile phones and random bits of cutlery and drawers and things. But still I keep going because it's worth it for the rewards. And yeah, I like to think that this very much is how academia can feel sometimes. I've tried to motivate myself. I've tried to keep a CV of failures along the way. So every job I didn't get, every paper I got rejected and grants. And I, quite ironically, Word refused to even save that once. some real-world examples as well. I'm sure you know of some as well, but so James Dyson, we know him as a very successful inventor, but actually he invented 5,126 vacuum cleaners before he found one that properly worked. And similarly, Henry Ford, at 28, he had great ambitions to become an engineer, and within 2 years, very quickly skyrocketed to chief engineer at Edison Illuminating Company. 5 years later, he built his own self-propelled vehicle, and that led to the founding of Detroit Automobile Company. But sadly, the company failed. He went bankrupt. But then, as we know, he then sort of went on and created his legendary Ford company. So now, a bit of a precursor, really, to the work we do in the lab, and I think where some of the future's likely to land as well. So I expect most of you will have heard of the Human Genome Project. For those of you that don't know too much about it, it was a project launched in 1990 and completed in 2003. And the aim was to identify and map all the genes in the human DNA. And so this is about 20 to 25,000 genes. And to determine the sequence of all the 3 million DNA base pairs that make up the genome. It was a huge undertaking, involved scientists around the world, private and public institutions. And what it ultimately achieved was a reference sequence that represents a mosaic of several individuals. And it's really enabled researchers to start to locate genes linked to diseases, understand genetic variation, and develop new tools. And this has really, really laid the groundwork for what we're now calling personalised medicine, so medicines tailored to an individual's genetic makeup. But this too was not immune to some failure along the way. There were early sequence limitations, so the technology at the time was really, really slow, expensive and error-prone. I think at the time there was great hope that we would find the gene that explained, every single disease, but what we've learned is that some disorders and diseases are much more complicated, like schizophrenia and alcoholism, and it's not likely just to be a single gene, but a combination of factors. Data storage and bottlenecks, the computing power at the time just wasn't sufficient to deal with the large volumes of data that this was generating. Genetic complexity, so that I think what we started to learn was that many genes and environmental factors come together and interact, so it makes it very, very challenging to unpick. There are annotation errors and gaps in the early drafts of the genome, so some missed genes, some that were coded completely incorrectly, and some overlooked entirely. And there was also ethical and legal confusion. There was a race between public and private entities. There wasn't a lot of regulation at the time around sharing of data, so people became quite precious, not wanting to collaborate because they wanted to get there first. And this slowed down public trust and ultimately policy development. So another one of my hobbies, I did promise to try and work in as many as I could. So this, as I said, this really laid the groundwork for what we're calling personalised medicine, which simply put is finding the right treatment for the right person at the right time. And an analogy I really like is one of my hobbies, growing and collecting cacti and succulents. It seems like a very specific hobby, but actually they grow in very random, unique places all over the world, and because of that they have very unique care needs. So they grow at different times of year, they need different types of light, different amounts of water, susceptible to different pests, for example. So they've all got their own regime. So that, I think, is a little bit, I suppose, about the concept of personalised medicine. This can be broken down further into what we call the 5 Ps. So personalised medicine can be used, for example, to predict somebody's propensity to develop disease. It can be used to perhaps identify and help prevent disease developing. Allows for more precise diagnosis, personalised and targeted interventions, and a more participatory role for patients. I think with more information, it allows people to for example, reduce trial and error in clinical trials and be more precise about that and understand likely side effects of drugs. So one of the probably leading examples of personalised medicine comes from spinal muscular atrophy, SMA, and this is an area that we've focused on quite heavily in the lab. So I'm going to walk you through a little bit about what we've done in SMA and what the future might hold. So SMA is a genetic neurodegenerative disease, and it's caused by mutations in the survival of motor neuron 1 gene. It affects lower motor neurons in the spinal cord and causes muscle weakness in the limbs and respiratory system. There are 4 main subtypes classified on severity, and approximately 1 in 6 to 1 in 10,000 live births are affected by SMA. So untreated, the most severe form of SMA type 1, children would typically not live beyond their 2nd birthday. They wouldn't be able to sit or hold their head. And then, as you can see on the diagram, the severity lessens with the subsequent types. So it's classified largely on age of onset and the clinical course that we know about. So I was very fortunate to come into SMA research where the green dot is, which was a really, really exciting time. So in 1995, the SMN gene was discovered as the causative gene for SMA. And very quickly, efforts moved very, very quickly to try and find therapies to treat or cure SMA. At the time, because it's simply caused by a mutation in the SMN1 gene, which leads to reduced levels of the SMN protein being produced. The idea was that, okay, if we can just put back that gene or increase the amount of SMN protein, that we can basically cure the disease. We've since learned that's not quite the case, but I'll tell you more about that soon. So I came in when there was quite a flurry of activity looking at small molecules and drugs that might help SMA patients. And then in the latter part of my postdoctoral years, actually the first preclinical gene therapy study came along. So this was a personalised medicine approach for people with SMA that targeted a very specific mutation. And in 2019 and 20, we saw the approval of several gene therapies that are now delivered clinically. So our early work was actually in moving on really from what I did in my PhD, was developing immunoassays to be able to measure increases in SMN protein in patient biofluids and cells. And these were intended for use in clinical trials, for example, to see whether particular drugs were able to increase the levels of SMN protein. And there are some examples given here of 2 drugs that were of interest at the time that since became less interesting. And I think from here then, because things move so quickly in the field, it opened up a lot of other possibilities and questions. So our research has evolved to look at understanding more about the molecular consequences of reduced SMN in various cells and tissues. The curious thing is that it's a protein that's expressed in every cell in the body, and yet certain cells and tissues and organs are more severely affected than others. So it's quite curious about why that happens. And to use this information to identify novel targets for future treatments, biomarkers to help monitor disease progress and responsiveness to treatment, and as I said, optimizing systems for testing drugs for SMA therapy. So I mentioned biomarkers in my previous slide, and this is where I think a lot of our work have moved towards, and other work unrelated to SMA. So for those of you that don't know, biomarkers are essentially measures of healthy or pathological processes in the body. And they might be imaging, such as images from CT or MRI scans, or molecular proteins, DNA, RNA and lipids. And biomarkers can have a range of uses, from diagnosis, prognosis prediction, finding new treatments, drug discovery and monitoring treatments. To identify biomarkers, proteomics technology that's really developed and really come along quickly in the last 10, 15 years is proteomics technology. So we've heard about the genome. The genes really carry the information inside the cell, but it's the proteins that are made from those genes that actually do the real work. So we've had quite an interest in understanding the proteome, which is the whole collection of proteins inside of the cell, and how those proteins interact with each other as well as work on their own, and which pathways they interact with. So to do this, we use mass spectrometry technology, and I won't bore you with all the technical details, you'll be pleased to know. But just to explain, I suppose, the difference between that and traditional biochemistry. So traditionally, we would usually need some sort of hypothesis or very specific question about a protein of interest that we would then have to go and look for. Whereas proteomics technology, mass spectrometry, allows us to simply go in and say, okay, what's there? How much of it is there? Without having any preconceived ideas. It has been referred to as a bit of a fishing trip sometimes, which is why you see the cartoon here. But actually, I think it's becoming more and more accepted as a very valuable tool for to support clinically applied research. And what it does is it allows us to ask questions like, are there proteins that are more highly or less expressed in a disease situation compared to control? And this can help us identify essentially what's going wrong at the molecular level in disease situations. So we've used proteomics. Some of our earlier work was looking to understand what SMN does inside of a cell and what it interacts with, what other proteins it works with. And the picture on the left is deliberately complicated to really illustrate how complex the situation is. So the green dot is the SMN protein, and every shape around it and line represents a protein that SMN is known to interact with at some stage and space inside of a cell. And we identified a number of these through our proteomics work and used bioinformatics tools to actually generate this image. But I think what it shows quite nicely is that, in theory, while you can increase the amount of SMN protein potentially using gene therapy or drugs, the downstream consequences of there having been reduced SMN could potentially impact on all of these proteins. And we don't yet know whether all of those perturbations are rescued then by the therapy that we're developing. And that's where a lot of interest has come in more recently, which I'll say a little bit more about. We also, around this time, we were also interested in finding small molecules that might be useful. And this was really before the gene therapy treatments had gained approval for use clinically. One of the reasons being, and which you'll hear more about, is one of the limitations of gene therapy is just how expensive it is. And so there is a great need for accessible treatments that are cheaper, essentially, and more easy to deliver. One of the studies I did in collaboration with Professor Tom Gillingwater and Tom Wishart in Edinburgh used proteomics technology to understand more about, at the molecular level, about how nerves and, I suppose in a mouse model of SMA, in terms of the differences in the nerve outgrowth profile. We found that a small drug from plants called quercetin was actually able to bring about quite remarkable rescue. So the bottom picture is a section from mice that were treated with quercetin, and you can see the long lines are actually the neurites that have grown through, and they look quite healthy, whereas the picture on the top, they're quite sort of scratchy, scarcely there. This was quite exciting at the time, and it was picked up by a number of newspapers and press. But shortly after, the gene therapy came along, and I think that the interest in small molecules remained to some extent for some of us, but I think had been clouded by the great excitement, for very good reason, around gene therapy. Some of the work we've done most recently, it's been led by Sharon Owen at the back and originally Daria Sholtic in the lab around SMA biomarkers. So one curious thing about the SMN gene is that we also, as humans, have a second gene, SMN2 gene. It's almost identical to SMN1, but it doesn't quite make up for the lack of SMN protein in SMA. And typically, and it was always thought to be the case, that the more copies of SMN2 gene you have, the less severe of a disease you would have if you have SMA. But what we've learned more and more over the years is that this often isn't the case, although there is some correlation. And it's really quite odd, and it led us to wonder actually what else is going on. Are there other modifiers of disease? So we've been using proteomics technology to try and understand a little bit more about the different types of SMA and whether the molecular profiles are similar across those different types of SMA, so that we can try and understand why somebody might have severe SMA and why they might not. So what we started with was 2 mouse models of SMA, one severe and one less severe model. We noticed in the literature there were a number of reports of SMA patients having cardiac abnormalities, and it was mostly reported for patients with less severe SMA, though we weren't sure whether this was just because patients were living long enough to be able to detect that. So we used quantitative proteomics technology to identify proteins that were changed in expression in those mouse models compared to healthy controls. We found a large number of proteins that were changed, and we looked across them to see if there were any commonalities across both models. We found several proteins, one of which was lamin AC, which was increased in expression. And this was quite interesting because mutations in the lamin AC gene cause a number of genetic conditions that are characterized by cardiac defects. So this helped us explain to some extent possibly some of the cardiac abnormalities that are seen in SMA. We wanted to know if these mice were then treated with the gene therapy that's similar to what's used in the clinic for patients, does this actually then correct some of those molecular changes that we see? And while some of the proteins were corrected back to the same levels as the controls, the lamin AC protein remained elevated. So what this suggests to us is that SMA patients, even though they're receiving gene therapy, they may benefit from cardiac health monitoring through the ages. And actually our own clinic at the Orthopaedic Hospital have started doing this now because of our findings. We've also looked to see whether we can identify any differences between cells from patients with SMA type 1, 2 and 3. And I mentioned before about the curious thing about the SMN2 copy number. So what we ended up with was a range of patient cells where the copy number actually did not predict the type of SMA that was classified clinically. So this was a really good baseline for trying to understand, okay, so why do these patients have severe SMA, for type 1, and why do others not have such a severe case? Because they're all caused by reduced levels of SMN, we expected there to be some molecular consequences downstream of that were conserved across the different types. But what you see in the heat map here is each coloured line represents a protein that was changed in expression. So from left to right, we have type 1, 2 and 3 SMA compared to their age-matched controls. And you can see that there's almost no overlap at all in terms of the proteins that were changed. And actually, when we used statistical cut-offs, we found that there were no proteins that were commonly dysregulated across all types of SMA. What this did reveal, however, was that some of the proteins we found might actually be useful as biomarkers to identify specific types of SMA. And it started to raise questions in our mind of, well, we've taken quite a simplistic view of treating SMA, and you'll see soon that that's had, made remarkable ways forward for SMA patients. But is it now time to start thinking about perhaps severity-specific treatments alongside of gene therapy that might help at different stages of life as well? So in terms of where we're at now, we have 3 approved gene therapies, nusinersen, Zolgensma and risdiplam, and I'll tell you a little bit more about some of the sort of limitations of these in a minute. But instead of me telling you more about how this actually affects SMA children, I'd like to share a short video with you. It's quite old now. It's from when the gene therapy was first approved, but I think it is quite remarkable. You'll see the transformation.
Spinal muscular atrophy, or SMA type 1, is the leading genetic cause of death for infants today. A devastating fact this family knows all too well.
Our first daughter, she was diagnosed with SMA type 1 at 6 months of age, and she passed away at 15 months old.
This is a disease that is devastating for infants. A large percentage of patients die before 1 year of age, but 95% 90+% are gone, unfortunately, by age 2.
SMA rapidly robs babies of their ability to move, talk, swallow, and eventually breathe. When Milan and his wife Elena found out they were pregnant with their second child, Evelyn, they prayed for a healthy baby. Unfortunately, that was not the case.
When she was born and the results came back and found out she was positive for SMA type 1, and we both kind of just broke down. We found out about the clinical trial here at Nationwide Children's Hospital.
This is really the first time that we've been able to apply gene therapy to any neuromuscular disease.
Today was a very special day for Evelyn. She was given genetic therapy for her missing SMN gene.
Babies with SMA are missing a gene vital for development. Through a one-time injection, gene therapy replaces this missing gene. For Evelyn, this gene therapy worked.
We start seeing changes as early as 2 months after treatment.
She started to push, and she started to get more active, holding her head up. And a little after 3-4 months, she rolled over on her own.
Now 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years, and she runs up to me and hugs me and says, "Dr. Mendel, I love you."
I forget that she has SMA. This is a healthy girl, and she does everything that a normal 3-year-old child would do.
Something like that has never been achieved before.
With our first daughter, it was just devastating to lose a child. You lose all dreams you had for their future. And now we can actually save for college.
So yeah, as you can see from that video, there have been huge advancements in therapy development and some absolutely incredible outcomes for many individuals with SMA. But this isn't the case for everyone. There are a number of key issues. So that there is a variability in response, and this is somewhat dependent on the SMA type, the age of treatment initiation, the number of SMN2 gene copies, and the lack of biomarkers as well to really stratify patients well. The timing of treatment is really, really important. So we know that the earlier that children get the treatment, they get much, much better outcomes. Newborn screening programs are available in some places but not universally implemented, and this often means that there's a delay in diagnosing children with SMA. And there are delivery challenges. Nusinersen is delivered via a lumbar puncture into the spine. Zolgensma is only available for young patients, and risdiplam is an oral medication that needs strict adherence and monitoring. And as I mentioned earlier, and you've probably seen things in the news, that the gene therapy carries a huge, huge price. So when it was first made available, it was the most expensive treatment ever to have been approved. So lots of regulatory things to work through as well with that. So in terms of the future, you've heard about some of the data sets that we've generated, but of course others are generating large data sets of, based on the proteome and genome studies. And it's very easy to do this now. Machines are much, much quicker and more sensitive than they used to be. So we've got these huge data sets, but what we've really lacked is a way of intelligently integrating all of these and the computing power to be able to do that. And I think with the new era in artificial intelligence driven, particularly AI-driven bioinformatics tools, I think this has really given us a bit of a boost and an ability to think about how we bring all of this together in terms of big data analysis and make the most of all the data that we've generated. So alongside gene therapy that's available now, I think some of the future will be around finding alternative drug-based treatments that could perhaps be used alongside of gene therapy to help further alleviate some of the symptoms. So it's important to stress, although the video I showed you was remarkable, it's a treatment and not a cure. We're living with these patients as they're going through the treatment in real time, so we don't know what the future holds for them. But we do know there are still some ongoing health concerns for many individuals. So our work here is not yet done. So just to wrap up a little bit before I move on to more education-related topics, just to say I've shown you about SMA today, but I've worked on a huge range of projects over the years using proteomics technology, from prawn crackers to pasties, sandfly saliva, sheep serum, many others, but some of the more neurologically based projects are listed here. So with Emily Story for her PhD and Sharon and Ian from the lab, we've done quite a lot of work to understand the molecular pathways involved in a rare childhood form of muscular dystrophy. With Monty Gates, who I remembered to name tonight, when I did my head of school introduction, I wrote an email to the school, said that I was married and that I have 3 dogs, and I named my dogs, but I neglected to name my husband Monty. So yes, hopefully I've made up for that this evening. So we've done work on Parkinson's disease to understand why dopamine neurons die and why they become more susceptible as we age to Parkinson's disease. Other work generally on nerve growth and stability mechanisms. Again with Monty, understanding how the corticospinal tract in the spinal cord actually grows during development. Some of these proteins and molecules are switched off once it's developed. So the idea is if we can understand these, we might be able to harness their potential for perhaps regenerating the corticospinal tract after spinal cord injury. Along a similar theme, we've done work again with Professor Tom Gillingwater and Tom Wishart in Edinburgh, understanding why some people's nerves repair more quickly than others after injury. And work understanding the side effect of drugs on cells. So one particular example is a drug called valproate, which has been used clinically for many, many years to treat epilepsy, among other things. But clinicians have noted over the years that patients on long-term treatment often have a risk of bone fracture. So we were interested in understanding whether proteomics could help us understand the mechanisms behind this. And in fact, we found that 2 key bone proteins were actually altered following valproate treatment. One of them is a protein that we know mutations in its gene cause osteogenesis imperfecta, which is a brittle bone disease. And so these findings helped explain the clinical symptoms that we're seeing, and that attracted quite a bit of press interest as well. And I think over the years, it's become, originally, proteomics technology was always thought of as I said at the start, as a bit of a fishing trip. It's not hypothesis-driven, it's a bit high risk, because you may find something, you may not. But what we're learning is it has huge value in terms of understanding the clinical impact of things and alongside of clinical trials. And a really good example of that is a drug that was trialled in 2006 for atherosclerosis. The trial was actually halted early, causing a $1 billion loss because of the side effects causing patient harm. In 2017, a team went back and reanalyzed the samples from patients that went through that clinical trial using proteomics technology and actually identified proteins that helped explain those side effects 3 months before they were actually apparent clinically. So I think that the use of the technology is becoming more and more embedded as, I suppose, state-of-the-art rather than cutting edge. So, moving from the lab to the outside world, this is where I'm going to focus now a little bit on my education-related roles at Keele and some of the insights I've gained along the way. And I think personalised medicine and personalised healthcare is a particular theme that runs through a lot of what I've learned over the last few years. In this sense, personalised medicine is, I suppose, can be seen as a bridge between worlds. It's not just about genomes and data, it's about understanding the whole person, the cultural, social and environmental context that brings us all together. So while I was Dean of Education, as Christian mentioned, we oversaw a number of programmes. And one of the developments we made and I contributed to was establishing a joint institute with Chengdu University of Traditional Chinese Medicine in China. And in 2003, we launched the institute, and we were very lucky to be invited over for a ceremony and to be shown around. What was really quite striking to me was the integration of cutting-edge technology, Western approaches to medicine with traditional Chinese medicine, and also the choice that people have over their healthcare. Just some examples here. There's me on the left having a traditional intervention called tweener. It's like a type of chiropractic massage. Some other similar methods in the middle, and then pictured as some traditional Chinese herbs. And then here you can see some really high-profile technology. So there's a medicines dispensing robot that automatically roves around the hospital, which was quite fun. And some of the examples we learned about were one was around genomic profiling for cancer treatment. So, for example, patients with liver or lung cancer may receive Western chemotherapy alongside of traditional Chinese herbs selected based on their genetic markers and profiles. And then combined with this, AI-driven diagnosis platforms help to fine-tune these combinations. AI is also being used to power traditional Chinese medicine diagnostic systems. And in the middle is my colleague, Adam Winterton, who's not here tonight, but he's got his head stuck in the machine and demonstrating how this works. But essentially, it uses the traditional Chinese approach of diagnosis using the tongue in terms of pulse analysis and how the tongue looks. And it combines this with Western results to cross-reference, for example, tongue colour and coating with blood test results. And from there, it will recommend personalised herbal formulas. We also saw some really impressive hospital-based integrated clinics where the patients are offered a combination of traditional Chinese medicine and Western medicine consultations. So, for example, a diabetic patient may receive insulin therapy while also undergoing acupuncture and herbal treatment that's tailored to their lifestyle. Some other examples now come from some field trips that we've been delivering. So, as I said, during my time as Dean of Education, I oversaw over 100 new programmes that we developed at Keele while I was chair of the Academic Quality and Academic Standards Committee. Some of these now sit in the new school that I'm head of. One of these programmes that doesn't sit in my school, but that was developed during my time as Dean of Education and still very close to my heart is the Masters in Global Health Leadership. And the premise of this programme really is about learning about healthcare systems around the world, comparing and contrasting, understanding that no one healthcare system is perfect, but actually if we can learn about what works really, really well in some situations and whether or not they're transferable to others, perhaps by bringing that information together and collaborating and working together, we might be able to piece together the perfect healthcare system. Alongside taught modules, the students go on field visits around the world, so we take them to 3 different countries to learn about the healthcare system there. So this programme was developed in collaboration with what's now NHS England and Professor Ian Cumming in the audience. And here is a picture of some of us outside the Ministry of Health in Rome, where I also managed to get myself locked in the toilet and needed a student to help me out, but I was slightly more graceful than when I got myself locked in the toilet in school. So one of the trips we went on was Thailand. We visited places very close to the border with Myanmar. And here, again, we see a rich culture of integrative medicine, bringing together Western and traditional Thai medicine. So Thailand has a dual healthcare system that blends Western practices with traditional Thai practices. And these include internal medicine, so herbal remedies tailored to individuals, spiritual medicine, so they might, for example, use tattoos and amulets, often personalised to somebody's birth data, and spirituality as well. And then all of these are brought together as appropriate to manage different conditions. A really nice example, I think, of personalised healthcare in Thailand was the approach to managing refugee and migrant care. There've been a lot of work and developments in this area, particularly since COVID. And there's a migrant health working group that supports, I think, close to about 300,000 migrants. It's staffed entirely by volunteers. I think there's about 7,000 trained at the moment. And they help bridge cultural gaps, so language barriers, for example. They help with disease surveillance, which is not necessarily straightforward with a mobile population, and healthcare education. And they also pioneer vaccine campaigns and health literacy programs tailored to specific migrant populations, including the Rohingya and the Karen tribal refugees, for example. So these are real examples of a personalised approach to public health that I think adapts the service needs to the culture and mobility of each migrant group. So while we were there, we visited a TB clinic. Here's us with our very well-fitted masks. And we learned about how the approach to TB was really quite personalised, I think, for the population that was being served. So there's actually a web-based application that supports personalised TB treatment decisions, and this uses a range of information including age, sex, location, medical history, any comorbidities that might impact on how the disease progresses, recommends safe drug combinations, for example, and predicts possibilities of side effects. And this has really helped the migrant health volunteers to identify TB symptoms within the community and refer individuals for diagnosis and treatment. And this often isn't easy. There's still quite a lot of stigma around TB. There's a hesitance to go and receive treatment because people would need to leave their families. They wouldn't be able to work and earn. So it's taken quite a lot of work and trust actually to get to the situation where they can make this successful. So they've really worked hard as a sort of cultural mediators in that case. But the programme overall really aims to reach, recruit, test, treat, prevent and retain people, and it allows a sort of personalised outreach as well as follow-up for mobile populations. The final major example is in India, another field trip we went on where there's an integration of Ayurvedic traditional practices with Western medicines and genomics. And here's some pictures from a factory we visited where they make traditional medicines. And here's one procedure which involves pouring hot oil over somebody's head and body. It doesn't look very pleasant, but I had a go, and I can assure you it was very nice. So this is quite interesting. It's a little bit similar to Thailand in the sense of combining Western and traditional medicines. But there are slightly different approaches, and it's tailored differently, I think, to the different population. Really impressed to see quite how advanced some of the healthcare systems, hospitals were in India in terms of technology-enhanced and personalised healthcare. And India is really leading the way in some respects. For example, some hospitals have AI-based diagnostics and chatbots. Here's one we visited, and you can see the pictures. Actually, it's a screen where patients can come along, they can select what their problem is, and they can actually choose from a menu which consultant they want. Very good developments in some areas around electronic medical record systems and personalised healthcare, I think, through some of these sort of custom checks and risk prediction tools. This is mostly so in urban situations, but I think there is an interest in work ongoing to try and reach some of the more rural areas. And at the moment, these are served by a particular role called an accredited social healthcare activist, otherwise known as an ASHA. And these are female health volunteers. They're taken from the village that they live in. And their local connection with the community really allows them to tailor advice based on cultural norms, dietary habits, local beliefs, and what they know about the population. And they have a really fundamental role in tracking histories, especially planning for things like maternal and family health, family planning, chronic conditions, and referring individuals to treatment where necessary, but also using traditional practices where perhaps it isn't necessary to refer somebody directly to a hospital. They too are using technology, AI-powered chatbot. It's called the ASHA bot. And this allows them to ask health-related questions in a variety of languages, Hindi, English, just as some examples. And it provides very context-specific answers straight away based on India's public health manuals. And one last example, Christian mentioned in his opening address about some of the work that I've been doing, actually led by Ian, sat here, and some other colleagues as inspectors, inspecting healthcare systems for the UK overseas territories. It's a bit of a stretch to try and shoehorn this in as an example of personalised medicine, but I think one of my career highlights was actually featuring on the front page of the Penguin News when we were in the Falkland Islands, so I couldn't resist the opportunity to put that in. But I think because of their size and population size relative and remoteness, then you can see that it's not really feasible to yet be thinking about developing particular personalised medicine approaches. But actually telemedicine and remote health consultations is really opening up this possibility for some of those areas by linking in with experts in the UK or other countries, for example. It is starting to become possible to try and tailor approaches for specific conditions, and chronic disease management is one of those. So, for example, hypertension and diabetes, I think it's becoming more and more feasible now to try and plan personalised care for those. So just to wrap up really, as head of school for the relatively newly formed Allied Health Professions and Pharmacy, I've obviously got a role with my colleagues in helping to shape the next generation of healthcare professionals who must be technology literate. You've heard lots of examples today, and this is not going away. They must be culturally competent. It's going to become more and more necessary to work across diverse global and local contexts and collaborate interprofessionally. And I think we're now at a stage where precision science really can intersect with human experience. So I think the future of healthcare education is about developing our students and future healthcare professionals to deliver care that's not only effective but also deeply personal. We're really lucky at Keele that we have some fantastic facilities. So you see some examples here of our simulated ward theatre. We've got simulation technology, for example, scanning technology. So we're really at the forefront in a way in terms of preparing our students for delivery of what healthcare needs now, but also in the future. And finally, just some thoughts really about what our students are going to need to learn and how we perhaps need to start thinking and shaping our curriculum. Technology is not going away. I think whole genome sequencing is going to become more and more widespread, so I think understanding what we do with this information. Other technologies such as wearables and apps, for example, are going to become more and more critical to healthcare and personalised healthcare. AI, of course, is not going anywhere, so it's about how we harness that and make the most of that in different contexts. And, of course, I've mentioned a lot through this about collaboration. None of the work that I've shown you here has been done in isolation. We work together all the time, and so I think keeping an open mind and fostering that as we go along is really important. So a few thank yous, of course, to my family for all their love and support over the years. Professionally, there are too many people to name, and if your picture's not on here, it doesn't mean I'm not grateful for your support, but these are just some of the key figures that have really supported me over the last, through my journey at Keele, really. So it's not an exhaustive list, but yeah, just to point out a few highlights. And, of course, our funders. So none of the research in our lab would have been possible without funding, which has largely come from charitable organisations. They're listed on the right-hand side. And last but not least, this particularly good boy who mopped up a few tears and the occasional red wine spill along the way. Thank you.